Hello.
Hi, how
How
are
are you?
you?
Good, good. So good to meet you. Thank you so much.
Oh, you're welcome.
So, as you might have noticed, I was at Texas A&M before coming to Purdue 4 years ago. So was the base. There he is. And I
Hi,
think he,
Cynthia.
yeah.
Hi.
Hey, Partha. Hey, Dan.
Hello,
Ben,
hello, Devesh. Hi,
Ben,
Dan.
Ben, if you can make me co-host, you can leave. I can take care of it now.
So you're saying we have a Texas A&M mafia going on here?
Yeah,
or-
looks like today. That was the word I was going to use.
I think everyone on this call except Ben is a Texas A&M in some way related.
Yeah.
So yeah, hello Devesh, hello Partha.
Hi,
Cynthia, I'm
Dan.
gonna, and actually I need to leave before this meeting is over. Devesh and Partha didn't check with my schedule before they scheduled this meeting this semester, so.
I'm so sorry, Dan, but thank you so much for your support.
I mean, you're supposed to, you're supposed to check with my schedule.
True, true, true. We kept the same 3:30.
I know, but I didn't know what was going on. But in any case, I'll be here for a bit.
Thank you.
Do you teach on Friday afternoons?
Me?
Yeah.
No, but I literally have meetings. You can go look at my calendar here real quick. I had one, two, if I include this one, 3, 4, 5, 6 meetings today. So,
Wow.
Very
You're
good.
administrator, Dan.
what's that?
You're an administrator, so that's what.
Well, no, it's a lot of class stuff, and because Capstone gets caught up in ABET, and we're doing ABET this year, so
Yeah.
I'm attending more meetings. But
Hi, Sid. How are you?
Said you're on mute.
You're on mute, Sid. Yeah.
Hi.
Yes, now I can hear you. Very good. Excellent.
Said you have a new dean who started today.
Yes,
So
yes.
I just
So a lot
texted him. Happy day one.
Yeah, so lot of news around. So yeah,
Yeah.
lot of news around. Yeah.
And Cynthia knows your dean well also.
Hi, Cynthia. This is Siddhartha from University of Maryland. Good to see you.
Good to see you too. Who's the dean?
Sam Graham, who was our chair. He's
Oh, yes, I do. Yes, I do. Yeah, you're lucky.
So, Cynthia, first of all, thank you so much. And also, so today Siddharth is going to sort of take care of today's session. So, Siddharth and I are sort of coordinating this semester and perhaps next semester this effort together. So, he's going to introduce you, and then you take over, and then introduce the speakers. So, we're hoping that we'll have good turnout today as well. So.
And do I need to make sure on their timing, or am I the person, or do you guys for moderator, do I control the time, or do you guys kind of control the time, or how do we do that?
I mean, typically, so far, the speakers kept to the time around 25 minutes, between 25 and 30 minutes. So far it was okay, but I think it would be great if you would like to keep a quick note, or maybe Sid is going to do that on your behalf.
I can take a quick
Yeah,
note.
Sid, you could probably.
And
do a little
it
warning
says
here.
25 for each, and then how long do we let the questions go on? I can't.
I mean, we will have a hard stop exactly at 5 p.m. Eastern.
Okay.
So you will say that, by a great time, just have more discussions. You can hang on with them, but later on, so we'll have
And
so.
people will, and I let them know to put both speakers speak, and then we go to questions, right?
That's
Yeah.
correct. Yes, that's
Okay.
correct.
And people put the questions in the chat?
Yes, they can put the chat. I can keep a tab of that, and if they want to go at the end, yeah, that's all.
So generally what we have done, if the number is more than 75, then we let them put that in the chat. If it is less than 75, they can turn on their mic and video, and they can introduce themselves and ask the question. So
okay.
And it's up to you, so whichever works best.
Yeah, so we're going to ask them to put in their questions in the chat anyway, and then whether or not we can ask them to unmute and tell it themselves, or Sid and you or I could kind of somehow speak out the questions, read out the questions. It's kind of the number as Devis mentioned. So it's like a little bit. Hi,
Okay.
Lihua, how are you? You're on mute.
You're on mute.
Yeah, I'm good. How are you?
Good, 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 good to meet you, and thanks for, of course, accepting our invitation to speak here today. I know that we only give very little time.
Yeah,
You
yeah.
know,
So it's
turnaround
it's
time.
within a 1 week notice. Everybody is just, yeah. That's
Yeah,
brilliant.
no problem. My
Yeah, so that's
abstract
our
and the
format.
talk.
And I know that Cynthia is smiling. Probably she got only few hours
Ah, yeah. Thank you very much. Yes.
overnight.
Just with everybody's permission, can I, like, when I start this, can I just spend 30 seconds introducing a faculty opening, like a multiple faculty openings in my department here? Okay, thank you. Thank you. I got pushed by Bala, that Professor Balachandran, our department chair, that said that I use this opportunity. There are, yeah, the professors are there. They will highlight that. So yeah.
Just ask
I
your
see
dean.
I have a postdoc opening then. If we're advertising here.
Absolutely, why not? As long as it's the time limit is 30 seconds for any of those.
Yeah, I mean, yeah. So if it, yes.
Absolutely. I think those are the ways to use this platform also.
Right.
Exactly.
Bala can send us a check for driving this, and we can have a named seminar series now.
Yeah, that's our payment, right? Is that we get to advertise. It's a advertising platform method.
So, Leo, do you want to check your slide just to make sure everything works
That
on your end?
sounds good. Sounds good.
Sure. So let me try to share my screen.
Yes, that would be great. Thank you.
Can you all see my screen?
Yes,
Yep.
wonderful.
Yeah. And I actually have some slides to intro, so I should probably double check too.
Yeah, that would be great. Please do that.
So, is that slides, or are you seeing the speaker view?
Speaker view.
Speaker
So I
view,
should
yeah.
swap
Maybe
it.
kind of swap it or duplicate something like that.
Is that right then?
Yeah.
Okay. Okay.
Hi, Siva. How are you?
Hi, Shiva. How you?
Hey everyone. Hello everyone. Yeah.
Yeah, good to see
Hey.
you.
Great to be here.
Shiva,
Yeah,
can you
so
quickly check your slides, maybe quickly?
yeah, that would be great.
Oh,
And
so should I
yeah,
just share my slides, you said?
Yeah. Just
just check it
share your
quickly.
slides and see if it's working fine.
Okay. Let's see.
And in the meantime, Siva, that you are sharing your screen, we just would like to say thank you because you only give few days of time, 1 week tops. So thank you.
No,
We're
absolutely.
just
No
joking
worries,
about
Partha.
that.
Yeah.
It's really a pleasure, and of course, I think it was sufficient time. And I'm sorry, I missed your first email. I'm glad you followed it up actually.
Yeah, we would like
Wonderful.
to thank you for organizing this and giving us the opportunity.
Yes, absolutely. Yeah,
Yes,
it's a wonderful way to learn work of so many people. I mean, I often look into the videos, and that's yeah. I mean, yeah,
absolutely.
it's
So,
beautiful.
Siva, would you like to kind of full screen view or something? It looks great. Yeah, just for the sake of it. Yeah.
Is it still okay?
Yeah,
Yeah.
perfect.
Great.
That's
Thank you.
perfect.
I would like to test one thing. I know I may have to switch. I have a couple of videos.
Yeah,
It's
please
trouble.
check
If
that.
it is trouble, I may not show them, but let's see. They do open because this is I have this in LaTeX PDFs, so sometimes we do have issues. Are you able to see the video?
Mm-hmm.
Are you able to see the video? Just a minute.
I don't think the video
Not
is
yet.
played on my side.
Okay.
No.
Yeah, it's static.
Let's see what we can get back to. Trying to get back to the share screen. Aha, here it is. Yeah, a couple of times I may have to do the switch, but maybe now you will be able to see it.
Yes, we can see the video now.
Oh, you can see the video?
Mhm.
Oh,
Yeah,
great.
it's like a pop-up.
I know, perfect. Yeah, I'll switch back and
So Siva, you could probably stop sharing your screen, and I think Siddharth would go.
Right.
I mean, Siddharth, you were going to show a couple of slides, right?
Sure,
Just brief intro, and then Cynthia, and then
Yeah.
I think.
Okay. Yeah. Yes. Absolutely. Absolutely.
Perfect. Thank you.
Okay.
So should we wait for probably 1 minute or something like that? Probably a couple of more minutes. I think a critical number is 25. We're almost there.
Are my slides visible?
Yes. You need to kind of, I think,
Yeah. Yes.
yeah,
They're little,
pull it
yeah.
away. Yeah. Probably we'll give 1 more minute or something and then get going. Oh, good. Speaking up. I think we could get started. Devesh, have you started the recording, right? Probably, or maybe this is a good time, right?
The recording has started. Go ahead.
Started. Okay, great.
Hello, everybody. Welcome to this week's Frontiers in Mechanical Engineering and Sciences webinar series. Before I start, I'm Siddhartha Das from the University of Maryland. I am one of the organizers of this very interesting series. Here is the full team, full organizing committee, if I may call it an organizing committee. So before I start about it, a very quick advertisement. There are multiple faculty positions, faculty openings at the Department of Mechanical Engineering, University of Maryland. The details are there on the web page of the department. If you need further information, please feel free to contact me or our department chair, Professor Balajan. Okay, with that, let's start. So today, our theme is a very exciting one: mechanics of soft and metamaterials. And we are extremely pleased that Professor Cynthia Hipwell of Texas A&M University has kindly agreed to serve as the moderator. And we have two speakers for this lined up: Professor Lihua Jin from UCLA and Professor Shiva Rudraraju from University of Wisconsin-Madison. A little bit about Professor Hipwell. Professor Hipwell is currently an Oscar S. White Junior 45 Chair 2 professor at Texas A&M University. She teaches classes on innovation and technology development, as well as leads the Invent Lab. Her research interests include nanoscale tribological and thermal phenomena at soft interfaces, including the finger-device interface for haptics and soft robotics. Professor Hipwell has been working in the area of technology development based upon nanoscale phenomena for the past 20 years. She received her BSME from Rice University and her MS and PhD in mechanical engineering from the University of California at Berkeley. Upon graduation, she went to work at Seagate Technologies Recording Head Division in Bloomington, Minnesota, to develop test equipment to characterize the interface between the head and the disk in hard disk drives. During her time at Seagate, Dr. Hipwell held various individual and leadership positions in the areas of reliability, product development, and advanced mechanical and electrical technology development. In these various roles, she established new business processes and an organizational culture that focused on developing innovative solutions, from root cause understanding, improved pace of learning, and discipline in experimentation and configuration management. She was inducted into the National Academy of Engineering in 2016 for her leadership in the development of technologies to enable areal density and reliability increased in hard disk drives, and was recently elected a National Academy of Inventors Fellow. So we are extremely proud and happy to have Professor Hipwell as the moderator of this series of this week's seminar series. So I would request Dr. Hipwell to take charge now. Thank you.
Okay. Thank you, Siddharth, and thank you for inviting me to be here in this seminar with you guys on this exciting topic. I'm very excited to hear the speakers, and why I just want to introduce a little bit. Why do we care about the mechanics of these soft materials? Why is this an important topic for us to be addressing? And soft materials are capable of large deformations, and they have properties which are similar to biological tissues. And this cool graphic here is actually from Dr. Jin's website, so we'll get to hopefully see more of that in her presentation. This is actually important for technology that interfaces with humans. So examples would be human-machine interfaces, for haptics, for remote touch, for being able to control human-machine interfaces with their body, for robots that work with humans so that they're safe and are able to interact with our soft tissues as well, for wearable electronics, so for devices that we're going to have interface with our body, and also for biomachines, so things that are inspired by biological phenomena or even self-emergent. So they're soft. Obviously, soft brings us a lot of exciting opportunity for technology, but there are a lot of challenges. We need to be able to do all the things that we do currently with hard, active, and electrical materials and structural materials with soft materials. So we need active materials. We need to be able to produce a large force, a large untethered force often. We need to be able to modify friction, temperature, optical properties, active with soft materials. We need to be able to make soft circuit elements. So the same types of conductors and semiconductors for wearable electronics. We have the soft interfaces, which is actually where our team is working, looking at how the soft materials interface with the different machines and how those machines interface with them. And then looking at emergent behavior for machines that are based upon biological systems and can actually be designed to evolve over time. So there's a lot of challenges here to be able to match some of the same type of performance that we actually have in our hard material space. And so that's why I'm super excited to see the talks today from these two dynamic young faculty. First, I want to introduce Dr. Lihua Jin, who is an assistant professor in the Department of Mechanical and Aerospace Engineering at the University of California, Los Angeles. Before joining UCLA in 2016, she was a postdoctoral scholar at Stanford University. In 2014, she obtained her PhD degree in engineering sciences from Harvard University. And prior to that, she earned her bachelor's and master's degrees from Fudan University in 2006 and 2009. Dr. Jin's group conducts research on the mechanics of soft materials, stimuli-responsive materials, instability and fracture, soft robotics, and biomechanics. She is the winner of the Haythorne Research Initiative Grant from the American Society of Mechanical Engineers, the Extreme Mechanics Letters Young Investigator Award, and the Hellman Fellowship. She's also a recipient of the UCLA Faculty Career Development Award and an NSF Career Awardee. So and as you see, she's going to be talking about soft metamaterials, which is really important in this active material space. And then Dr. Shiva Rudraraju is an assistant professor in the Department of Mechanical Engineering at the University of Wisconsin-Madison and a fellow of the Granger Institute for Engineering. He leads the Computational Mechanics and Multiphysics group at UW-Madison, and his research interests are broadly in computational modeling of mechanics and multiphysics-driven microstructure and morphological processes in materials, structural, functional, and biological materials. He was nominated to the Gallagher Young Investigator Award and received the Faculty Scholar Award from the Granger Institute for Engineering. He was also awarded the Madison Teaching and Learning Excellence Fellowship. His current research projects are supported by ONR, ARO, and NSF. He received his PhD in Mechanical Engineering and Scientific Computing from the University of Michigan at Ann Arbor. So with that, I'd like to turn over the slides to Dr. Jin, and so we can hear about soft metamaterials. Welcome.
Thank you very much for the very nice introduction and also a nice overview about the opportunities and challenges in the field of soft materials. Today, I'm mainly going to tell you more of our recent progress on shape-morphing and energy-absorbing metamaterials. Mechanical metamaterials are materials with microstructures, which can give rise to unusual mechanical properties that are difficult or impossible to achieve in homogeneous materials. Researchers have achieved mechanical metamaterials with negative Poisson's ratio, high stability, and also twisting behavior and compression. Mechanical metamaterials are largely enabled by 3D printing techniques because their properties are governed by geometry, which give us a vast design space. And sometimes mechanical metamaterials are also called architected materials. So today, I'm mainly going to talk about some of our recent progress on developing metamaterials for new functions. The first example is about spatial temporally shape-morphing metamaterials. Then I'm also going to talk about reusable and absorbing metamaterials. Let me start with the first example. What you see here is an octopus. As we know, octopus are entirely soft machines. As you can see, they can significantly deform their own body and squeeze in and out of a narrow glass bottle. You also notice once it gets out of the bottle, it senses the background color and can immediately camouflage itself. So what's the mechanism of the camouflage? Well, that's because the octopus can spatial temporally change their skin texture, and moreover, they can also change their skin color because there are bags of pigments underneath the skin. Using muscle, they can stretch to show different colors. Of course, this is very challenging to mimic, but the first question we want to ask, is it possible that we can spatial temporally even change the texture of a surface? To achieve that, there are a few challenges. So first of all, we want to select a unit cell which can show temporal behavior. Then moreover, this temporal behavior needs to be easily tuned by geometry or materials. Then we can combine those unit cells with different temporal behavior, then we can see a pattern evolution. And moreover, if we can further tune, assemble and reassemble, disassemble, reassemble those unit cells, we can see different patterns. Here are the unit cells we select. So we select a viscoelastic shell as our unit cell. What you see here is that we are indenting this viscoelastic shell. If I immediately remove my indentation, the shell is supposed to immediately pop back. So it is showing a monostable behavior. However, if I hold my indentation for a certain amount of time, as you see here, it does not immediately pop back. But you need to wait for a certain amount of time and then eventually pop back. So this behavior is called pseudo-bistable behavior, such that it's a fake bistable. It can remain in the deformed shape or the inverted shape for a certain amount of time, but then after you wait for some amount of time, it will pop back. Therefore, we choose this as our unit cell and bond it on a box and seal them, and then we can pneumatically actuate the deformation of the unit cell. And moreover, very soon, I'm going to show you that by tuning material and geometry, we can significantly tune this snapping behavior. Therefore, by combining this kind of unit cell together with different temporal behavior, we can pattern them into different surfaces, and those surfaces can also evolve as a function of time. Moreover, we can assemble, disassemble, and reassemble into different patterns. Before I show you the experiment, let's first establish some theory to understand this behavior. So this is the shell geometry, and it is subject to a pressure difference. And we hold the pressure difference for a certain amount of time, then plot the corresponding displacement, W here. As you can see, the displacement does not immediately recover to its undeformed shape, but it has to remain for a certain amount of time and then eventually recover its original shape. So the time we hold the sample, we call it T creep, and the time we have to wait, we call it T snap. And here we conduct both finite element analysis and also viscoelastic shell theory to analytically analyze, both computational and analytically analyze this problem. To understand the mechanism, let me first plot the instantaneous pressure displacement relationship for different geometry. Here, D represents the shell thickness and R represents the radius of the shell. As you can see, the pressure displacement relationship first increase, then decrease, and increase again with the displacement. For some of the curve, it actually intersects with the x-axis, indicating a bistable behavior. That means, for example, for this intersection point here, that means even when you remove the pressure difference, the shell can stay in that displacement. So therefore, for thin shells, they tend to be bistable, but for thick shells, they tend to be monostable. So that is the instantaneous behavior. But even when you have some monostable shell, if you indent it and hold for a certain amount of time, you can shift this curve to change it from monostable to bistable. That's why you observe this pseudo-bistable behavior. But if after you remove the indentation, the shell will recover from bistable back to the monostable, such that after a certain amount of waiting time, it will snap back. So this explains the mechanism of this viscoelastic shell. Then moreover, we can quantify this snapping time, and we see it can be significantly changed by how long you hold the sample, by the geometry, and moreover by the material. Here, G represents how much relaxation your material allows. Then once we understand, we can build the unit cell and test the unit cell individually and also in an assembly. Because this temporal behavior of the shell is intrinsically controlled by its material and the geometry, therefore, in an assembly of those shells, we can use a single control by connecting all those shells together with a single control of sensor, valves, and pumps. Then we can simply control this complex spatial temporal behavior by a single control. As you see here, I'm showing you 4 different types of shells by tuning the geometry and also selecting 2 materials with different viscoelastic property. We first extract air and hold for a certain amount of time. Then you see the second row pops first, after that third row, and finally the fourth row. And moreover, as long as we design the structure with a big enough box, we observe that the behavior of an individual unit cell and also in an assembly are similar. Then once we have all those components, we can pattern them together to achieve spatial temporal surface evolution. So what you see here is 3 by 5 unit cells, and we first extract air, and then after a certain amount of time, we let it go. So you first see a number 5, and then you see number 6, and finally you see number 8. As you can see, those patterns can evolve as a function of time. We further demonstrate temporal patterning of emoji. Again, we first extract air, hold for a certain amount of time. Then we remove the load. You first see a smiley face, then a wiggling face, and finally a stunning face. With such kind of surface, with tunable surface texture, we can actually achieve many other functions such as tunable friction, wetting property, and optical properties. Then next, let me also show you a spatial temporal control of friction. So we pattern our surface with different unit cells. Then we build a setup to measure the surface friction. And we design the surface in a way that when all the shells are in the convex state, the friction is low. But when they're in the concave state, the friction is low. When they are in the convex state, the friction is high. Therefore, we can measure the friction as a function of time. When we start, all the shells are in the convex state, such that the friction is high. Then we extract air and the friction become low. Then we remove the load. As a control, here the blue curve shows the case with all the shells with the monostable state, in the bistable state, such that once you remove the load, it's bistable, therefore they can stay in the concave state and the friction remains low. But the black curve corresponds to the case where all the shells are in the monostable state. Therefore, once you remove the load, all the shells recover its original shape, showing a high friction. But now the red curve corresponds to a mixture of different viscoelastic shells. As a function of time, you can see some increase of friction, and then after a certain amount of time, increase of friction, and then increase again. Therefore, you can spatial temporally control the friction. Okay, now let me switch the gear a little bit to talk about energy absorbing materials. The traditional energy absorbing materials make use of plastic deformation or fracture to dissipate energy. So they can have high energy absorption, but they're not reusable. So here, our idea is make use of snapback buckling of wide columns to achieve reusable energy absorption. So recently, we discovered that if we tune the width to length ratio of a column, they can show very different buckling behavior. So if you compress a column, when the width to length ratio is low, so what you observe is a traditional Euler buckling, or here we call it continuous buckling. So that means after the buckling points, if you gradually increase the displacement, the force gradually increases with the displacement. However, if you increase the width to length ratio, you observe this snap-through buckling. So that means after the buckling point, the force actually decreases with the displacement, showing a negative slope. If you further increase your width to length ratio to some extent, you observe this new snapping back behavior. So that means after the buckling point, both the force and displacement decrease along the equilibrium path, with the slope still remain positive. We can make use of such snapping back buckling to achieve reusable energy absorption. So that means if you along this force displacement curve to this state of point A, if you keep increasing displacement, it has to snap to this state A2. Correspondingly, the energy also has a jump. And the energy difference between these two wells corresponds to the energy dissipation. So if you further unload it, the sample will snap back from the state A3 back to A4. So therefore, a column exhibiting this snapping back buckling is capable of dissipating energy in a reusable, rate-independent behavior because all the deformation are elastic. It's completely recoverable, and also it's self-recoverable, which means after unloading, it completely recovers its ordinal shape. Therefore, we want to stack such wide columns into a multi-layer structure with each layer an array of wide columns to achieve energy absorbing metamaterials. So here is how we fabricate this material. We use a multi-material 3D printing together with sacrificial molding. We first print this mold with composed of PLA and PVA. Then we pull pre-cured silicone elastomer and cure them. Then eventually we dissolve water-soluble PVA and then left with this metamaterial. And finally, we also need to add some hinges as constraint, such that we can achieve the deformation modes we want instead of some global buckling. Here I'm showing you the behavior of this metamaterial. So when you load the sample, you can observe 8 peaks here corresponding to the buckling of these 8 layers. And you observe a long force plateau. And during unloading, again, you observe 8 peaks corresponding to unbuckling of the 8 layers. And the loading and unloading curve show a large hysteresis between them, indicating energy dissipation. And once you unload the sample, it can recover its ordinal shape immediately, indicating reusability and the self-recoverability. Then in order to understand this zigzag loading and unloading curve, we further develop some theory to predict the behavior. So we start with the force displacement curve from a single column. Then we can simplify this multi-layer structure as a chain of columns connect in series. So therefore, the external force equals to the force in each of the column, and the external displacement equals the summation of the displacement of all the columns. Then, given an external force or given a force in a column, it can have 3 different possible displacements. Now, if you have a chain of columns, if we do this combinational problem, we can have many possible displacements corresponding to all the branches you observe here. However, not all the branches are stable. We further need to examine the Hessian matrix to determine the stable branches, which are indicated by those solid curves. Then you can either just connect the solid curves, or if they're discontinuous, you just connect in the neighboring one. Then you can determine the loading and unloading paths, and the area in between them corresponds to the energy dissipation. Here I'm showing you the loading and unloading paths for columns with different width to length ratio and a different number of columns stacking together. And as a result, we see that the energy dissipation of snapping back columns is higher than the snapping through columns here, shown here under the same n, and grows faster with n. And here I'm further comparing the experimental result and theory together. As you can see, they agree relatively well with each other. And please note that there's not a single fitting parameter in this theory. And you may ask, what is causing the deviation? I can tell you that's merely because of the imperfection in the experiments. Then further, we conduct drop tests to examine the energy absorbing property of those material. So we throw the sample from a certain height to mimic an impact from the bottom, and then we measure the peak force at the top. So here I'm showing you the measurements, the peak force as a function of the different amount of energy impact at the bottom. As you can see, the peak force reaches a plateau in a wide range of external energy corresponding to a force mitigation region due to the column buckling. And moreover, we notice this peak force actually agree very well with the peak force we measure in static test, indicating the rate independency on a strain rate across a very wide range. And moreover, if you notice in the video, after the impact, the sample almost doesn't bounce back too much, indicating energy dissipation in this process. Because this is metamaterial, the behavior is governed by the geometry, we can easily tune the geometry to tune the property. For example, here by tuning the width to length ratio such that the columns are in the snapping through regime, we can observe there's very small hysteresis between loading and unloading, especially if you plot it together with the previous case. You see the energy dissipation is much lower. Moreover, we can apply a preload to the sample to tune the peak force and even the stability. So we add a preload by adding up weight to the sample. Now, during the loading, you still observe 8 peaks. However, during the unloading, you notice the sample does not recover its original shape, but it is locked in the deformed state. So therefore, in this example, I showed you making use of snapping back buckling of wide hyperelastic column, I can achieve reusable energy absorption. However, because the property comes from hyperelastic material, the amount of energy absorbing is limited because those materials has low stiffness. Then we need to think about, is it possible that we can achieve high energy absorption capacity, but still maintain the reusability? Then we need to think about different energy dissipation mechanisms. Those can be reusable and with no damage of your material. Then we come up with the idea of making use of friction of materials. And we design here the granular metamaterial by integrating granular materials and elastic components for energy absorption. we came up with 3 different designs. The first one is cylinders bound by elastic bands. The second one is spheres threaded by elastic strings. And moreover, we can also achieve spheres wrapped by elastic membranes. Let me first show you a case of cylinders bound by the elastic bands. So in all those metamaterials, those granular particles, they can slide between each other to dissipate energy. But on the other hand, because of those elastic components, they can maintain the integrity of the structure and eventually can even recover to its original shape. So what you see here is during deformation, those cylinders can slide with each other to dissipate energy. And therefore, here we have 6 layers, and in the corresponding force displacement curve, you observe 6 peaks corresponding to the collapsing of each layers. And moreover, because the collapsing behavior is governed by the materials, constitutive materials, therefore by tuning the elasticity of the elastic components, we can tune the peak force and even the order of the collapsing behavior. Moreover, I'm also going to show you the performance of the metamaterial through impact test. So on the right-hand side, what you see is this metamaterials with 8-layer structure. But on the left-hand side, as a control, we show the metamaterial in the fully deformed state. And at the bottom, you see a single supported glass and the materials in between to protect the glass from an impact. Now, as you can see, right, for the control experiment, the glass is completely broken under high impact and scattered everywhere. But on the right-hand side, you can see, even with a high impact, the glass remains intact because the metamaterial can dissipate a lot of energy for the impact. Finally, I'm also going to show you another spheres threaded by elastic strings. In particularly, we can completely design to different symmetry. The left one, we call it interstitial orthogonal tetrahedral symmetry. And the right one, we call it dense orthogonal cubic symmetry. And you compress this metamaterial and unload it. And as you can see, we can tune its force displacement curve. And more interestingly, you notice that when we unload, the sample on the right-hand side can completely recover, and sample on the left-hand side cannot, because the friction trapped it in the deformed state. So therefore, all the properties can be tuned. So now, let me summarize my talk. I first talk about design of spatial temporally programmable shape-morphing metamaterials. Then I'm also showed you 2 examples of reusable energy absorbing materials, making use of snapping back buckling of white columns and the friction between granules. What's more important is that those are all metamaterials. Their behavior can be widely tuned by changing the geometry and the material designs. With that, I would like to thank the group members who conduct the work, thank the collaborators, funding support, and thank you with your attention.
Thank you so much. And so for those of you who have questions, we'll be taking questions for both speakers at the end of the 2 talks. So you can put them in the chat, and we will probably let you actually ask your question live, but go ahead and put them in the chat for queuing. And then I'd like to next have Dr. Rudraraju come on and present his talk looking at the emergent structures.
Excellent. Thanks a lot, Cynthia. I know that you gave us a great beginning by sort of setting the stage for the need for these understanding sort of the mechanics or deformations of these soft structures, both in active and inactive materials and also in the biological context. And I'd like to begin by thanking the forum, the coordinators, Partha Mukherjee, Siddhartha Das, and Devesh Ranjan, and others. This, as a young faculty, or as an early career faculty, this is a great opportunity. And thanks for everyone. And by the way, it seems already difficult to follow Lihua Jin's great talk. Very, very interesting and fascinating material. But I'll try my best. So with that, share the screen here. Okay. Let me get this out of the way. Excellent. Okay. Thanks everyone. Thanks to all the audience, all the students and faculty in the audience. So today I'm going to talk about essentially certain classes of mechanical and mechanical plus chemistry driven phenomena in biological membranes, again soft materials, and and our approach to modeling them and trying to get to some emergent phenomena in this process. And I'm Shiva Rudraraju at the University of Wisconsin-Madison. And just a brief introduction to the work of my research group. On the physics side of our interest, my group's work is broadly classified into applications in material science and manufacturing process on one side, and applications on biophysics. And it's more or less a 50-50 split. And the biophysics part really excites me. It's everything is emergent phenomena there. And really what enables me to do this work is this great team I have. So everything I'm going to show you today is really the hard work of my graduate students, Debabrata Odhya and Rahul Gulati, and Shaoshan Zhang, who's a postdoctoral fellow at University of Michigan, who's collaborating me with this work. And the real drivers and the people who provide the inspiration and sort of provide experimental evidence and sort of even insights are my great collaborators, Padmini Rangamani at San Diego, Krishna Garikapati at Michigan Ann Arbor, locally Christian Frank at UW-Madison, Ashfaq at Texas Arlington, and I'm more recently part of an ONR group called Panther, which is a very big project looking at traumatic brain injury and the neuronal damage which happens. So that brings in a great set of collaborators, and I'm still trying to establish those connections. So I'd really like to thank all of these people who make this work happen. So with that, let me motivate this talk. Why am I interested in biological cells and why membrane phenomena? Again, biological cells, of course, they're sort of the basis of all living systems. And any one of you who has, had a run-in with any biophysics at the cell scale knows the cells are a universe in itself. They have tremendous complexity, tremendous richness in terms of the physics, and and that physics leads to great amount of mathematics, if one cared to understand. And there are various geometric structures within a cell. Of a particular interest to me are the membranes. Because membranes are very interesting because they're really two-dimensional structures embedded in 3D. And when you study the deformation or transport on membranes, it's much more complex, and it's at least in my opinion, it's very elegant, those kind of problems. And given my background in finite strain mechanics, I'm more interested in cell scale phenomena that occur on the membrane, more specifically, the phenomena which are driven by mechanics and chemistry. And here I have a collage of 3 different problems I'll be talking to you today. On the top left corner is the filopodia protrusion, which is one of the mechanisms by which cells walk. On the top right is one of the mechanisms by which cells open channels, open gates for material to flow in and out. That's the piezo-1 protein activated dome formation. And a very exciting application is on the bottom left. That's of endocytosis. And I'll make a small connection, but a very strong connection to, how this is essentially what's causing havoc with the world in terms of the COVID virus. So making these three the central applications for today's talk, I'd set the mathematical context for this work. Biological membranes, again, on the top, you would see this cartoon of how a biological membrane looks. They're extremely thin membranes, but they're actually fluidic membranes. They're really not fully solid. They have a much more fluidic behavior or viscoelastic behavior. And they have a complex set of these lipid bilayers and with hydrophobic, hydrophilic protein distributions. And it's a very dynamic environment. But for the problems of our interest, we are not interested in all that immense complexity. We are interested really on the mechanical stiffness of the membrane and how it modulates the morphology or the shape. So to do that, the very first challenge is, it goes to 101 mechanics or kinematics, is how do you represent the geometry? Traditionally, we would use a Euclidean basis, but as I said, membranes are really two-dimensional manifolds embedded in 3D space. So a 3D basis like XYZ would really get us into numerical or mathematical complications. And the natural setting is to use a coordinate-free description or a curvilinear coordinate system, wherein essentially the basis advects along the shape. That is really the natural structure for these natural setting for these structures. So we adopt that, and in that I'm providing the reference and current configurations, which are the non-deformed and deformed configurations for a generic membrane. And a couple of slides on how we set this up, because the whole goal of this part of my work is developing a mathematical model, a numerical model, and then applying it to different problems. So that the core of this is the standard curvilinear coordinate setting, wherein we have a curvilinear coordinate system. The basis of that are the tangent vectors, and from the tangent vectors we get to the primary geometric quantities of any membranes, which are the metric tensor, and through the second fundamental form, the curvature tensor. What the metric tensor and the curvature tensor, essentially they are equivalents of strain and curvature in more regular mechanics. What they let you track the geometry and see how the geometry is changing. And more, we get to special invariant forms of those, which are the mean and Gaussian curvature, H and K. So these two curvatures, really, it's just by tracking the change in these two curvatures, one gets to know how the geometry is evolving. And the specific kind of kinematics we induce is called Kirchhoff-Love shell kinematics, very ancient and classical kinematic theory for surfaces. The assumption is because the membrane is very thin, the out of plane strains are neglected, and it's really, we only treat the in plane strains, which could be found from the metric tensor and the curvature tensor. And again, these are highly simplified forms of this. If I were to expand any of these, probably they would expand out to a few lines, if not a few pages, as you'll see next. So with that setting of the geometry, we jump into the governing equations. There are two kinds of governing equations broadly. As I said, we are interested in mechanochemical phenomena, which is mechanics and chemistry, sometimes individually acting, sometimes acting together. Mechanics sometimes induces chemistry, and chemistry modulates mechanics. So we are interested in mechanochemical phenomena, it's a coupled multiphysics. So at the ground level, we are really interested in, first, what you're looking here is the governing equation for surface mechanics. On the top is the momentum conservation equation in terms of the stress and the moment, and the P is the body force and T is the traction vector. I don't want to get into any of these derivations. Really, we assume something called a hyperelastic setting, and starting with a well-known form of energy for these membranes called the Helfrich energy, we derive sort of the stress and moment terms, and all of this is completely differential geometry. That's the setting for all these derivations. And really, in the context of membranes, this was first done by Saur and all very recently, actually, and they developed this proper treatment, which really was my point of departure to adopt these models. And likewise for chemistry, again, the most simplified, the essential ingredients are how the mass is, diffusing in terms of this this is a representation of a surface representation of the Fickian diffusion model. And again, what you have there are the surface fluxes and the chemical potential coming from the free energy. So again, I could get into any amount of these details, but roughly with that math, that was the first step, the mathematical characterization of these processes. The second step is to put them in a numerical framework. And the central challenge with membrane modeling, as some of you may know, is the continuity requirements. They have to be continuous. You cannot have, sort of kinks and other things. So selecting the correct numerical basis is very important. And we adopt a more recent evolution in the finite element analysis, not so recent, maybe at least for a decade, called isogeometric analysis, where essentially the bases are splines, which give us a high fidelity control over the curvature and the fields on that curvature. So we adopt that framework and we implemented everything we do is in-house codes. We develop our own codes and implement everything in-house. And then we use a lot of mathematical recipes to get around problems. One is, these are very higher order partial differential equations. So I'm sorry if I forgot to mention that. All of these are at least fourth order partial differential equations. So given these higher order partial differential equations, there are difficulties enforcing boundary conditions. So we go with something called the niche procedure. And then if one were to expand the governing equations I have, it would be at least a few pages long, and one is bound to make mistakes. So we adopt more state-of-the-art techniques of automatic differentiation to essentially automatically generate our set of governing equations. So with that numerical setting, I'll move on to the problems. But before, this is the summary of essentially what we do with various biophysics problems. We take any given biomembrane phenomena. We first understand sort of the mechanics and chemistry, processes, and then define it as a boundary value problem. Essentially, what are the boundary conditions, the load conditions, the time and spatio-temporal evolution, and we identify the kinematics, the transport, and the mechanics process, and then put all of that in a variational formulation. Then take this into the computational framework of isogeometric analysis, and then comes the hard work. The students, they have to run all these simulations, do essentially parametric studies to understand these problems, or then figure out various phase diagrams. So with that come the applications I'm going to present today. And here I'd like to take a minute's break and emphasize the goal of, it's not a purely numerical interest in these problems. A central challenge with modern biophysics is a lot is known qualitatively. People know phenomena A leads to phenomena B leads to phenomena C. But when one tries to precisely quantify them, that's when sort of you're opening a can of worms, because when we try to model, we cannot say A goes to B somehow. We model partial differential. We need precise boundary conditions, precise parameters. And when we put that in, we realize, A really doesn't lead to B. So if I were to summarize our motivation is to quantifiably, verify and validate what is qualitatively known in the biophysics community. And we adopt that principle and apply it to all these three problems and a couple of more problems later. And sort of these results I'm presenting are in these references for those of you who are interested. The first problem is that of membrane tube pulling. So cells have this membrane, which is the outer layer, and the membrane actually stretches out. And it's really that stretching mechanism, and it forms these tubes, like Philopodia-like tubes. And that's how cells have a lot of processes which are enabled. Most importantly, that's how cells walk. If you ever looked at bacterium walking, it forms these tubes and it really uses them as anchors to walk. The way these structures biophysically manifest is you have the membrane and you have a huge scaffold of proteins which push the membrane out, hold the membrane at a given height, and sometime in some other phenomena, they have to truncate the membrane out. So we wanted to understand this process. And the very first step in every problem is identify its mechanics and chemistry and define its boundary conditions. Here, the boundary conditions. We essentially have the surface representation, and we apply various displacement, angle, moment, and traction boundary conditions. And all of these numbers are either from literature or experimental evidence. And then we try to validate do these numbers make sense for this specific phenomena? So here we're doing two things. We are pulling the membrane and we are applying a constriction pressure. And very quickly into the results, what you see is, when you apply pressure, the membrane pulls up. But this is one of the simplest problems in membrane physics. So we have an analytical solution on the top right. We validate the result with analytical and axisymmetric solutions. But ours is full 3D. We don't have any restrictions on symmetry as against more traditional axisymmetric methods. So we do validate against existing understanding of force displacement for these. But now, having validated this, we are able to do much more richer problems which are not accessible to analytical or axisymmetric methods. A good example of that is this problem, which is essentially membranes, when they are stretched for certain processes, they have to be pinched. Pinching is a way to to chop the membrane, and proteins do this. They try to package membranes by chopping them. So we took the mechanisms which are understood, applied the loads, and then we found something very fundamental in this problem, which is now published in Soft Matter. The classical understanding was it's an axisymmetric pinching, and proteins have to apply a certain force. And after a lot of study, what we realized is, there's a buckling-like instability, a structural instability in these geometries. And if membranes were to attack those instabilities, they could get the job done with 1/10 of the energy spent. And so that's sort of what these pictures show, that the axisymmetric solution is symmetric. Whereas when we apply the full 3D solution, you see that it's no longer symmetric pinching, but it has this more, flattened pinching profile. And another thing, we found that it matters where you pinch. The protein can decide to pinch this sort of tube-like structure at the cap or at the tube location or the base location. It really turns out it's all about the location. The proteins have to assemble at specific locations, and they can really get their work done with 1/10 of the energy spent. And we validated these numbers both in terms of how much pressure is required, and where are the instabilities and what are the correct points for the proteins to attack. And of course, this is all numerical predictions, and we are behind experimental. We are trying to get some experimental validation of that. And if I can briefly show you one small simulation of how this happens. So as you're probably seeing, this is a membrane which we are applying a symmetric load, and you'll see it flattening and collapsing. And essentially, there's a lot of protein scaffold which is providing the forces which are not represented here. There is the collapse. But this is happening at 1/10 the force, whereas it would have taken much more force if it was a symmetric collapse. So back to the talk. And a second problem of interest is essentially these biomembranes have channels which have to be opened and closed. Essentially, that's how material gets inside and out of the cells. And one of the channels is called a piezo 1 mechanism, wherein mechanics, the application of forces on the membrane, really closes and opens these membranes. And it's beautiful configurational change process. If you're looking at the figures A and B, B is a flattened figure where you have these 3 arms which make up the protein. And under certain conditions of mechanical loading, these proteins, 3 arms actually rotate, and they flip, they pop essentially the membrane out. And the popped state is A, which is the activated state. So the protein, by popping and unpopping, can go between these activated, deactivated states. On the bottom is the geometric representation of this process. And the piezo dome is where essentially the protein accumulates. And this is again the boundary conditions. I don't want to get into that. And what we are able to show in this is we are able to quantify under what surface tensions or mechanical loadings we are able to get a good amount of gating response, opening and closing of those channels. The hole at the center is the channel. So essentially, how much amount of work the cell has to do or the proteins have to do to let material inside and outside. So this is a parametric study with respect to the surface tension. So given short of time, this is the more exciting problem. I promised to make a connection to sort of COVID. This is a very central mechanism in all cellular processes called endocytosis. This is essentially how material gets inside a cell. You just can't walk in a cell. You have to request the cell to open the gates, and it gives you a special packaging to save it from the immune response of the cell. And then the packaging takes you inside into the nucleus. And what better, you know, in the bottom you have a virus, and this is every day what COVID virus or any virus tries to do. So what is our interest in this is we want to again take the conventional understanding of endocytosis and see, is it really, what are the forces involved in this dome-like structure forming, and how is this packaging happening? So again, we quantify, it took us a long time to quantify these boundary conditions. And the coat area, that red area, is where you have curvature proteins which accumulate and create that shape. And that's how they really, and the virus or sort of nutrients come in and sit in that sort of that bump, and then they close the hole. So we did a couple of very detailed studies, and Shoshan took the lead on this. What we did is we studied the effect of those protein curvature on how the shapes are generated. Again, here we say that yes, there is a symmetric shape evolution, but there are pathways of instability wherein you can pop into unstable structures and, you know, take advantage of those instabilities, and you're able to package much better. All of the, probably 1,000 of these simulations are captured finally into this phase diagram, where we are able to show that there is an area of symmetric and asymmetric sort of packaging available. And proteins can, depending, you know, they may prefer to get into the asymmetric side, which is essentially selecting the right amount of area they want to recruit on, and the right amount of curvature they want to induce to produce those shapes. And they have beautiful 2-fold, 3-fold, and 5-fold symmetries which come out of this. With that, I'd like to, you know, this is the last slide. What do we have here is, this is the fourth problem of interest, and where all my entire group's biophysics, all hands on deck for this problem, which is we are now trying to model the mechanochemistry and electrostatics of neurons. We are looking at single neurons, and we are asking our question. One is a more sort of a regular question, which is how much load can a neuron take, and when does it fail? So this is a very detailed viscoelastic modeling of the neuronal membranes and the, you know, surrounding material. But an equally important problem, we all know that neurons conduct electricity, but we are trying. It's mostly, you know, done through something called a Hodgkin-Huxley model, which got the first Nobel Prize in biophysics in the 50s. But it's an ODE model. It says a great deal about the mechanisms, but again, there are a lot of missing details today about how electrical signal jumps through something called a myelinated part. And if that jump doesn't happen, we would be more like, you know, our IQ would have been 100 times lower than what it is. So we are trying to look at the electrical conduction using various processes and trying to answer that question, that how is the signal conduction happening so fast, and how is that related to the mechanics. So I only have a very basic simulation at this point to share with you. So as you probably see, this is essentially the electrical signal passing through along the axon. And what are represented are potassium ions, sodium ions, and a lot of chemistry is shown in that plot. Anyway, quickly, I want to end the talk. So with that, I'd like to summarize. And what I had tried to show is our effort at developing a robust numerical framework for modeling mechanics and multiphysics of membranes using the framework of Kirchhoff-Love kinematics. What we have already achieved is we have modeled 3 different problems: membrane pulling, piezo-1 dome, and endocytosis. What we are currently working on is understanding electrostatics and mechanoelectrostatics in neurons, and another process called phase transformation on JNT unilaminar vesicles. What we have established is that yes, in the problem of membrane necking or membrane pinching, they are favorable energy barriers which could be used to, you know, get the job done by the proteins. And also in the process of endocytosis, we obtained a phase diagram and showed that there are symmetric breaking pathways which are more favorable. But as I said, biophysics of membranes is enormously complex. We only have some amount of physics representation. We are trying to add more and more. What is in progress now is adding viscoelasticity, more surface transport, coupling with electrostatics, and eventually contact modeling. With that, I gratefully acknowledge the funding for this work from ONR and the Wisconsin Alumni Association at UW-Madison. And thanks again for this opportunity, and I'll be open for questions.
Okay, thank you. Thank you so much. So now I invite the audience to either raise their hand electronically and we'll call on them, or post in the chat questions that you have for either Dr. Jin or Dr. Rudraraju.
I have a question.
Okay, go ahead.
Yeah, wonderful talk, both of you. You really illuminated the session. Excellent talks, both of you. Thank you very much. I have a question for Professor Rudraraju. So, Shiva, when you make those models, do you account for the fact that there is an electric double layer of charges around the membrane on either side, both on the side that is exterior to the cell as well as the cytosol side, the interior to the cell?
Excellent. So yes, so there are two, it's a great question and something troubling us, because there are two directions of electrical charge, ionic and charge electricity conduction, which is the transverse and the longitudinal. So
Right.
currently we have a very good representation of the transverse direction. So we have, yes, charge distribution outside and inside. And we have the chemistry, because that's the more famous Hodgkin-Huxley and the later models. So we know that part. The central challenge, and which I think is open, I don't think people enough know, is how does this transverse induce longitudinal? Because eventually current goes longitudinal. That coupling is what we are trying to understand. To answer your question, yes, we do have representation on either side.
Okay. And do you, and so when the follow-up to this, so when let's say you are considering interaction of, let's say, a nanoparticle with a membrane.
Yes.
The fact that as the nanoparticle gradually approaches, and like particularly, let's say, for non-specific adhesion, where you don't have some kind of a receptor ligand kind of interaction.
Sure.
And there is an actual kind of a warping happening.
Absolutely, yes.
There will be a dynamic evolution of this exposed charge, because typically, let's say, if you have metal nanoparticle, metal is a product, it cannot support charge on its surface.
Yes, yes.
The moment it comes in contact with the charging that is there pre-existing on the membrane, that part is no longer charged.
Yes, yes, yes.
So what happened to that ions? Because iron has to be conserved at that location.
Absolutely.
Between the metal and the membrane, there is no ions.
Yes, yes.
So is your model accounting for this corresponding electrohydrodynamics of the ions?
Yes. So at this point, no, but that is what we are trying. When I said we are trying to extend the model to surface transport, the central challenge is the ionic part of the PDEs are well known, but we have to couple that with the change in curvature.
Right.
The curve, it comes through the surface operator, which actually evolves as you iterate.
Right.
So while the maths of it is clear, what is not yet clear to me, at least, is how is that coupling happening? Wherein there is something like a reaction term in the charge which is coupled to the curvature. That specific term is not known. So what we plan to do is cook up some term. I believe we can model it, but we don't know exactly that term, frankly.
So there is one, please feel free to stop me here. I mean, maybe I'm just going on here. So if there are other questions, please feel free to interject me. There's a wonderful paper by Professor Howard Stone, published, I think, a couple of years back in Biophysical Journal. So as you know, Professor Stone from Princeton is one of the biggest authorities in this low Reynolds number transport.
Yes.
So he looked into this, and he looked into this corresponding electrohydrodynamics of the ions.
Absolutely.
Okay. So there's a fluid flow might also occur there, because you are creating an ionic gradient, and that kind of ionic gradient will create what is known as diffuse osmosis.
Yes, yes, yes.
So there will be localized flows, very tiny localized fluid mechanics, very like a micro nanoscale fluid mechanics going on over there. Nobody has ever thought of this.
No, actually, you're almost getting me excited to jump on that problem. Of course, I didn't know this particular work of Professor Stone. I know his other works, but I'll definitely look it up. And it's a very beautiful multi-physics problem, as you're saying.
Yeah, I published quite a few series of applied physics letters on this, but doing a very simplistic 1D membrane
Sure.
and the nanoparticle interacting with the EDL induced by the 1D membrane and the nanoparticle. I will
I
send
definitely follow up with you. That I'd like to know more. Thanks
Sure.
to that.
Thank you. I stop here.
Okay. Other questions? Yes.
Yes.
Go
I see.
ahead.
Yu Huang, yeah, go ahead. Yeah.
Yeah.
Yuhan Hu from Georgia Tech. I have a question to Lihua. Lihua, wonderful talk. Good to see you here. So just two questions. The first is regarding to the first part of your talk. So how did you experimentally realize these different time scales in making these elastomers? And what is the range of time scale that you can tune in the material system? And so the second question is regarding to the last example you gave. It's very interesting and it brings a lot of design space. So I think you have maybe ongoing work in looking into all these different geometries and materials. I just have a special just a wonder. If you change those beads to material that can generate plastic deformation, so usually that provides another layer of dissipation mechanism. But that I think will also suppress your instability or delay it in certain way. Have you thought about whether that would help with this energy absorption, or in this case, will not be helpful?
Yeah, thank you, Yuhan. Thank you for your question, and it's also very nice to see you here. Coming back to your first question, you ask how we can tune the response speed. We select different types of materials with different viscoelastic properties. Actually, those materials are not that different on the viscoelastic response time, but they are very different on how much they can relax. So for one material, maybe it can only relax 10%, but another it can relax like 30, 40%. Then we can also tune geometry. It's actually a little bit tricky because the system works the best if you are close to the monostable and bistable boundary. So it's a little bit sensitive. If you tune geometry too much, then you are very far away from the boundary. But there is some regime that we can also tune in the geometric regime to achieve different behavior. Then, yeah, coming back to your second point, indeed, we are still have some ongoing work considering particles which can even undergo some plastic deformation or even combining different energy dissipation mechanism. I think, as you said, if you even actually, as you see, right, even in our current system with friction and elastic system, by tuning the geometry, we can actually achieve either stable behavior or monostable behavior. Right. So in some system, you have enough friction to trap it, lock into the deformed state, but in some system, it can bounce back. So we have not carefully studied the further contribution of plastic deformation, but I think you are probably right. It could also potentially make it harder to recover. But it could be very interesting questions to try to answer in the future. Thank you.
Thank you, Lihua.
Other questions? If other people don't have one, then I'm going to jump in because I have one. So very interested in the friction modification. What is that? You mentioned that there isn't as much variation in the relaxation time. What do you think is the limit of how fast you can get it to modify, and what kind of drives it? Is it just that viscoelastic property, or what can drive it to faster switching?
Okay, thank you for the question. So am I allowed to share the screen again?
I think so.
I stopped sharing the screen. Yeah, you should be able to.
So actually, because all this behavior is completely determined by your indentation. So as I said, if you apply indentation and without any holding, so the response time is actually 0. So it can instantaneously bounce back. Therefore, how much time the T-snap is determined by how much time you hold your indentation. The longer you hold, the longer time it takes for it to snap back. As I also explained the mechanism. So essentially you have an initial curve, but this is instantaneous curve. But now it's viscoelastic. If you deform and you hold it, you can shift this curve to different extent, right? So the longer it deviates from the initial state. So therefore it takes longer for it to recover the initial state, to change back from the bistable state to the monostable state. So therefore, if you want the response to be fast, then you just hold for shorter amount of time. Then it will snap back relatively fast.
So when you say, yeah, that's the relatively that I'm, because remember, I'm like usually at nanoseconds and things like that. So is 0, what is 0 really? How fast is that 0? That's kind of what I was.
So essentially, if instantaneous, it's in the elastic limit. So therefore, it will instantaneously recover. Of course, there are some inertial effects, so that, and some viscoelastic effect, but you can consider as an elastic system. So, for example, right, this kind of system, it's monostable, therefore it can instantaneous snapback. But of course, in all those elastomers, unpreventably, there is some viscoelasticity. It may slow down a little bit, but in principle, it should be pretty fast.
Okay. And the other question I had is, how small have you actually made the structures to be like for the octopus skin, for the little deforming ones that come up through there? How small have you made them, and what do you think the limit is of where you could go with that?
Yes, thank you for the question. So far, yeah, I'm not going to show my screen again, but if you check our scale bar, so far for our unit cell, the radius is around 1 centimeter. So I would say it's pretty large. Actually, I have to say in all the 3 metamaterials we show, they are in a centimeter region. But now we are also trying to develop 3D printing to fabricate metamaterial in a much smaller scale. Hopefully, in the future, we can achieve much smaller scale.
Okay, and what do you predict the property change will be as you scale it? Do you lose, like, sometimes when you scale down some of these softer things, you lose force? I mean, what are you predicting would happen as you scale?
Right, because right now we are studying viscoelastic system. So therefore, there are no intrinsic length scale in the problem. If you scale it down, say, probably 10 micron, probably no problem. However, if you further scale it down, and because it's soft material, there could potentially be surface energy effect to some extent. Yeah, I would predict if you go to micron, yeah, especially if the based on the elasticity and surface energy, right? You can also define length scale. So that will be the critical length scale below which you will see surface energy effect.
Okay, thank you.
Thank you.
Other questions?
I think if not, probably Professor Hipwell, I think we could potentially conclude here.
Okay, yes, thank you, everybody.
Thank you.
So,
Thank you.
thank you. So I think from all of us in the organizing committee, a big thank you to our great speakers, excellent 2 talks. And a big thank you to Professor Hipwell for your generosity in time and being with us today. This is also an opportunity for our, of course, speakers to interact among themselves, and also, as you know, with Professor Hipwell here. You have found a great moderator here and to discuss your further. And also with Siddharth, I think you got some good discussions with Rudraju. So keep doing that. That's our goal here. And if Siddharth, if it's all right and the base, so probably you could conclude our session here.
Yes.
And we'll stay tuned for the next announcement shortly.
Thanks everyone. Thanks
Thank
again.
Thank you.
Thank you.
Thanks for
Thank
the
you.
organization.
Absolutely.
Yeah,
Thank you very much.
so much.
Bye-bye.
Thank you. Bye-bye.
Bye.